Hey there! If you've seen my last video, you already know how GPCRs work up to the point where the G-alpha subunit activates adenylate cyclase. Well, this video picks up right from there. We're going to take a closer look at what adenylate cyclase does, how different types of receptors control it, and most importantly, how this leads to the activation of PKA, or protein kinase A. Adenylate cyclase is a membrane-bound enzyme that converts ATP into cyclic AMP, or cAMP, a vital second messenger inside the cell. This molecule acts as a messenger, triggering the next steps in many signaling pathways, including the PKA pathway we'll explore next. But did you know that adenylate cyclase can be both switched on and off depending on the receptor it interacts with? When a ligand binds to a receptor coupled with the stimulatory G protein, G-alpha S, adenylate cyclase is activated. This means it ramps up the production of cAMP. On the other hand, when a ligand binds to receptors coupled with the inhibitory G protein, G-alpha I, adenylate cyclase activity is reduced, producing less cAMP. X-ray crystallographic analysis has pinpointed the regions in G-alpha S.GTP that interact with adenylyl cyclase. This enzyme is a multi-pass transmembrane protein with two large cytosolic domains, each of which is a catalytic domain that binds ATP and converts it to cAMP. cAMP, produced by adenylyl cyclase, activates protein kinase A, or PKA, which phosphorylates various intracellular targets to mediate diverse cellular responses. Inactive PKA is a tetramer made of two regulatory and two catalytic subunits. Each regulatory subunit blocks the catalytic ones using a pseudo-substrate domain that mimics real substrates but doesn't get phosphorylated. Each R subunit has two distinct cAMP binding sites, called CNBA and CNBB. Binding of cAMP by an R subunit of PKA occurs in a cooperative fashion. That is, binding of the first cAMP molecule to CNBB lowers the KD for binding of the second cAMP to CNBA. The catalytic subunits travel into the nucleus to change gene expression. Inside the nucleus, these catalytic subunits phosphorylate a transcription factor called CREB, or cAMP response element binding protein. CREB sits on specific DNA sequences called CREs, or the cAMP response elements, found in the promoters of cAMP-regulated genes. Phosphorylation of CREB at serine 133 allows it to recruit a powerful coactivator called CBP-P300. This coactivator links CREB to RNA polymerase II and other gene regulatory proteins, thereby stimulating gene transcription. That was the nuclear response. But the real question is, how does a single hormone molecule, like epinephrine, lead to such a massive cellular response? The answer lies in signal amplification, a brilliant strategy that cells use to multiply a signal at every step of the pathway. The specific example here is the pathway depicted in this figure. Binding of a single epinephrine molecule to one G protein coupled receptor induces activation of several molecules of adenylyl cyclase, the enzyme that catalyzes the synthesis of cyclic AMP, and each of these enzyme molecules synthesizes a large number of cAMP molecules, the first level of amplification. Two cAMP molecules activate one PKA, but each activated PKA phosphorylates and activates multiple target proteins. This second level of amplification may involve several sequential reactions, in which the product of one reaction activates the enzyme catalyzing the next reaction. The more steps in such a cascade, the greater the signal amplification possible. We've seen how cAMP and PKA amplify signals and even change gene expression. But there's more. PKA doesn't just stop here. The next video will uncover how it rewires metabolism, especially glycogen breakdown.